So this model is called BDFaCSMedloy, so this is a model that we acquired in November last year. So it wasn't much used yet because after the COVID, students didn't have opportunity to start with the instrument, but hopefully next month we will have a PhD student starting his project on this machine here. And what it does is it measures the cells and particles that are in lipid suspension, and they pass through a beam of light that's inside. It's got a laser inside. You can analyze blood cells, like red cells, white cells. You can analyze carcinogenic cells. And for the analysis we're going to be doing next month, they're going to look at bacteria in water, treated water. So they want to look at the populations of bacteria in that water sample. And then we can send it for DNA sequencing to see which bacteria is on that sample. So the main reason is after you treat the water, so it's safe to drink, but there's still some bacteria in there. So we want to look if there is something in there that actually shouldn't be in there. Like for example, you know that some bacteria are resistant to antibiotics. So we just want to make sure that after the water is treated properly and there is nothing harmful for humans. So this can be used by Agrifood and environmental students if their project needs this kind of analysis. So at the moment I know that it's used country-wise for like cancer research. Also, as I mentioned, for looking at bacteria or other microorganisms in water samples, for example. And so it's used globe-wide because it's a very powerful tool for that kind of analysis. Because it can analyze like very small particles. So it's like 0.5 micrometers. So it's very small particles. So you can have, for example, a water sample in here. You put it in this tube. You put it inside. So you then press load sample, the sample will go up, and then you press start analysis. So it gives you the number of events, and then when you are satisfied with your parameters and if you are satisfied with the data, you just press record, and then you will have a graph like this, so with dots. So basically it tells you that we have two different types of bacteria in that water sample, and then you can separate them, so you can like draw gates around it, and then you can divide the populations into two. So you had like initially you had like just this group of dots, but you can divide them into two. So once you have that, you can tell the machine to sort them into two different tubes. So you would put this rack here, you put it inside, and then what it will do is like for P1 it will go to one side, and for P2 it will go to one side. So P1 will be population 1, and P2 will be population 2, and then you can send that for DNA sequencing, so you can get it, basically the genetic code for that bacteria. You put the tube in that bit, it goes up, and the machine has got a detector inside, and what it does, like we have different types of filters, so depending on your study, you select different types of filter at the beginning of the experiment. So in here you can select how many filters you would like to have and the parameters. And then when you are satisfied with that, you press record data. So as soon as you record data, you're going to see this graph. But at the beginning it doesn't make much sense because the bacteria is grouped, but you can kind of differentiate that there are like 2 or 3 different populations. So what you do is like you draw gates, and then you can select different parameters in the graph. So like you can plot that with different like exponential or logarithmic. And then once you're satisfied with that, and you can actually see that you, like here, that you have 2 different populations. You draw a gate around them, and you just tell the machine which one is going to be population 1 and population 2. And then what the machine does, it's going to put population 1 in tube number 1 and population 2 in tube number 2. And then you just press sort, and then it just sorts into those 2 different tubes. Yeah, you put them there, you close them, and then when you press sort, you will collect them, and then you got 2 different populations of bacteria in 2 different tubes, and then you can do DNA analysis with that. You can do DNA sequencing, or depending on what you're doing. So it's technically the same for red cells and white cells. You could do that. If you had like a blood sample, you could do that. And the way also the machine works is we have antibodies that link to specific cells, and in these cases they will be labeled with fluorescent dyes. So in the case of DNA, we need a dye that binds with DNA so we can see it, so the machine won't be able to detect that if the sample that we have, for example, a water sample, hasn't got any dye inside the liquid. Another option for this instrument is, rather than having these two collection tubes, you can also split into you can use a 96 well plate. So in this case, like if you have population 1, 2, 3 and 4, you can tell the machine that you want those populations divided into X number of wells, and then you can also use that for your DNA analysis or DNA sequencing.